Hello everyone, welcome back to our YouTube channel Pharmacy by Swati. Today, we will start a new subject that is Pharmaceutical Analysis of pharm first sem. And the lecture will include definition and scope of pharmaceutical analysis, different techniques of analysis, methods of expressing concentration, primary and secondary standard solution, preparation and standardization of various molar and normal solutions such as oxalic acid, sodium hydroxide, hydrochloric acid, sodium thiosulfate, sulfuric acid, potassium permanganate, and ferric ammonium sulfate. And the remaining topics will be covered in the next lecture or part B. So, let's start the lecture. Pharmaceutical analysis is a branch of pharmacy in which we identify, determine or analyze the quality and quantity of any particular drug or sample. And pharmaceutical analysis involves a variety of techniques which is used to identify and quantify the active ingredients, impurities and degraded products present in pharmaceutical formulations. And the main purpose of pharmaceutical analysis is to identify substances, purify them, separate and quantify them, and also determine the molecular structure of chemical compounds. It also ensures that drugs are safe and effective for use by the patients. And there are two types of pharmaceutical analysis, that is qualitative analysis and quantitative analysis. Qualitative analysis involves identification, and these tests are performed to indicate whether the substances or compound is present in the sample or not. Simply, it determines the presence of any particular substance in drug or sample. Tests such as limit test, detection of gas evolved, formation of precipitates, color change reactions, melting point and boiling point are used to identify the presence of any substance in the sample. Quantitative analysis. It involves estimation. Techniques are used to quantify any substance or compound in the sample. It includes the quantity or amount of substance present in the particular sample or drug. Now, our next topic is scope of pharmaceutical analysis. Pharmaceutical analysis involves in the identification of nature of drug or active pharmaceutical ingredient. It helps in the analysis of raw material. To become a pharmaceutical analyst. In food industry, testing of quality and quantity of sample and also includes the packaging of product. Pharmaceutical analysis also helps in the diagnosis of any disease such as HIV is observed by ELISA or enzyme-linked immune sorbent assay method. It has also scope in the cosmetic preparations such as lipstick creams, nail paints, shampoos and conditioners. In environmental studies such as to check the pH of rain rivers and water resources. And analysis of different environmental factors such as temperature and humidity. Next, an important topic is different techniques of analysis. Mainly, there are 4 types of techniques of analysis. These are chemical method, electrical method, instrumental method, biological method. Chemical involves various titrations such as acid-base titration, non-aqueous titration, precipitation titration, complexometric titration, and redox titration. and these titrations are well explained in the next unit. Electrical method. It includes potentiometry, conductometry, polarimetry, and voltammetry. Instrumental method involves gas chromatography, thin layer chromatography, high-performance liquid chromatography, high-performance thin layer chromatography, and biological methods such as cup plate method and turbidimetric method. Now, our next topic is method of expressing concentration. So, first one is molarity. Molarity is defined as the total number of moles of solute present in the 1 liter of solution. It is denoted as capital M and its unit is moles per liter. Molality is defined as the total number of moles of solute in 1 kilogram of mass of solvent. It is denoted as small m and its unit is moles per kilogram. And normality is the number of gram equivalent per liter of solution. It is represented as capital N and its unit is equivalent weight per liter. Weight percentage is defined as the weight of solute divided by weight of solution multiplied by 100. And volume percentage is the volume of solute divided by volume of solution multiplied by 100. And parts per million is the mass of solute in grams divided by mass of solution in grams multiplied by 10 to the power 6. Next topic is primary standard. Primary standard is a reagent having the known concentration and used to find out the concentration of another substance or sample. Properties of primary standard solutions such as these are highly stable and less reactive. Highly pure and its purity is about 99.9%. Molecular weight of primary standard solution is generally high, and it has no water of hydration. For example, molecular weight of potassium dichromate is 294.19 gram per mole. Next one is secondary standard. It is a standard that is prepared in the laboratory for a specific compound or element analysis. It is made from primary standard solution. Properties of secondary standard solutions such as these are less pure as compared to primary standard solution. Second is standard solution are hygroscopic in nature. Concentration of secondary standard solution is unknown. Standardized against a primary standard. And these solutions are more reactive than primary standard solution. For example, sodium hydroxide and potassium hydroxide. Now, our last topic is preparation and standardization of various molar and normal solutions such as oxalic acid, sodium hydroxide, hydrochloric acid, sodium thiosulfate, sulfuric acid, potassium permanganate, and steric ammonium sulfate. So, first one is oxalic acid. Its method of preparation is about 6.3 gram of oxalic acid is weighed accurately and dissolved in distilled water. And then, make up the volume to the 1 liter in volumetric flask. And the standard solution of oxalic acid, which is a primary standard solution, is used to find the strength of sodium hydroxide and potassium hydroxide, which is a secondary standard solution. Next one is sodium hydroxide. For one normal solution, we need to calculate the equivalent weight of sodium hydroxide, which is 40. To make one normal solution, dissolve 40 gram of sodium hydroxide in water and make up the volume of 1 liter. And for zero point 0.1 normal solution, 4 gram of sodium hydroxide per liter is needed. And its method of standardization is 2 grams of potassium hydrogen phthalate weighed accurately, which is previously powdered and dried at 120 degrees Celsius for 2 hours. And dissolve it in 75 milliliters of carbon dioxide-free distilled water. Then, add 0.1 milliliter of phenolphthalein solution and titrate with the sodium hydroxide solution until a permanent pink color is produced. Note that each ml of 1 molar sodium hydroxide is equivalent 20.2042 gram of potassium hydrogen phthalate. Next one is hydrochloric acid. Its method of preparation is dilute 85 milliliters of concentrated hydrochloric acid in water to produce 1000 milliliters solution. And its method of standardization is weigh accurately about 1.5 gram of anhydrous sodium carbonate, which is previously heated at about 270 degrees Celsius for 1 hour. Dissolve it in 100 milliliters of distilled water and add 0.1 milliliter of methyl red solution. Then, add the acid slowly from a burette along with the constant stirring until the solution becomes faintly pink. Now heat the solution to boil and cool and then continue the titration. Heat again to boil and titrate further as necessary until the faint pink color is no longer affected by continued boiling. Note that each ml of 1 molar hydrochloric acid is equivalent to 0.05 to 99 gram of sodium carbonate. Now, next one is sodium thiosulfate. Its method of preparation is dissolve 25 grams of sodium thiosulfate and 0.2 gram of sodium carbonate in carbon dioxide-free distilled water and then make up the volume with water about 1000 milliliters. Its method of standardization is take 10 milliliters of potassium iodate solution and then add 2 grams of potassium iodide and 5 milliliters of dilute sulfuric acid. Now, keep it in dark for 10 minutes. Then, add 2 to 3 drops of starch indicator and then titrate with sodium thiosulfate using starch solution as indicator until the blue color is disappeared. Next one is sulfuric acid. Its method of preparation is dissolve 54 milliliters of concentrated sulfuric acid in 600 milliliters of water, cool and then make the volume to 1000 milliliters with distilled water. And its method of standardization is take about 3 grams of anhydrous sodium carbonate, which is dried previously at 270 degrees Celsius for 1 hour. Dissolve it in 100 milliliters of distilled water, then add 0.1 milliliter of methyl red as indicator and titrate with 1 molar sulfuric acid with constant stirring until the solution becomes faintly pink. Heat solution to boiling. Cool and continue the titration until faint pink color is no affected by continue boiling. Next and second last compound is potassium permanganate. Its method of preparation is dissolve 3.2 g of potassium permanganate to 1000 milliliters of water, heat on a bath for 1 hour, allow to stand for 2 days, and then filter it. And its process of standardization is take 20 milliliters of oxalic acid solution and add 5 milliliters of 1 molar sulfuric acid and heat at 700 degrees Celsius. Then, titrate with potassium permanganate solution taken in the burette. And end point is the appearance of pink color that persists for 30 seconds. Last is ceric ammonium sulfate. Method of preparation is dissolve 65 grams of ceric ammonium sulfate with the aid of gentle heat in a mixture of 30 milliliters of sulfuric acid and 500 milliliters of water. Then, cool, filter the solution. And process of standardization is weigh accurately about 0.2 g of arsenic trioxide, which is previously dried at 105 degrees Celsius for 1 hour, and transferred to a 500 milliliters conical flask. And then wash down the inner walls of the flask with 25 milliliters of an 8% weight by volume solution of sodium hydroxide, agitate to dissolve, and add 100 milliliters of distilled water and mix. Add 30 milliliters of dilute sulfuric acid, 0.15 milliliter of osmic acid solution, and 0.1 milliliter of ferron sulfate solution and slowly titrate with the ceric ammonium sulfate solution until pink color is changed to a very pale blue, adding the titrant slowly towards the end point. Note that each ml of 0.1 molar ceric ammonium sulfate. So, we have completed first part of Unit 1. If you have any queries regarding our lectures, then ask in the comments section. Thank you, keep supporting, and take care. Hello.